respected session chair, respected professors, and all my dear friends. I am Niranjan Koirala, a PhD student in Sunmun University, South Korea. The topic of my presentation today is biotransformation process for the enhanced production of sakuranetin, genkwanin, and rhamnetin, along with full-fledged utilization of glycosyl transferase, possess the compound as a potent pharmaceutical and industrial agents. So I want to start with the introduction to flavonoids used in this research. So the flavonoids are these are basically plant secondary metabolites which are produced by the plants during the stress situation. These have different properties like antibacterial, antimicrobial, anti-cancer, anti-inflammatory, and many other, to name a few. So I have chosen 3 different compounds, sakuranetin, genkwanin, and rhamnetin, for my research. So sakuranetin is a 7-methyl-naringenin, which is a chiral flavonone present in rice, finger root, yerba santa, spike pepper, and Populus diabetina. Similarly, genkwanin is one of the major non-glycosylated flavonoids found in some herbs, such as guenca floss, rosemary, and the leaves of Citrus laurifolius. And rhamnetin, or naturally occurring 7-methyl quercetin, is abundantly present in Rhamnus petiolaris, coriandrum, Syzygium, and Prunus serracus. So like having so much of benefits from these compounds and flavonoids, but still we are not able to utilize fully due to its low oral bioavailability and low water solubility and less stability. So like we have tried to do some modifications to these 3 different compounds so that we can increase the stability and solubility so it can be utilized in a better way in the market. So like metabolically, this is clearly due to instability and very low oral bioavailability of the dietary flavonoids. This can be attributed to free hydroxyl groups of most polyphenolic compounds, resulting in very rapid conjugation by glucuronidation and sulfation. So our technique to increase the stability of these compounds is methylation, which after doing which the free hydroxyl group in flavonoids dramatically increase their metabolic stability and enhance their membrane transport, leading to facilitated absorption and greatly increase oral bioavailability. So the strategy which we have used in this research, the outline of this research strategy is shown here. This different compound naringenin, epigenin, and quercetin, 3 different substrates which we have used. This is the E. coli host which we have engineered by using SAOMT. This is the Streptomyces avermitilis derived O-methyltransferase, which can methylate at the 7 position, this position, this position, and this position. This is this one is EC SAM synthase. This is adenosine methionine synthetase, which will help to increase the cofactor of SAM cofactor inside the E. coli. So after this feeding in the E. coli in vivo system, we could produce sakuranetin, genkwanin, and rhamnetin, which are methylated at the 7 position in each case. So these are the different strains which we have used in this study. So after the production of these compounds, we check the HPLC, high performance liquid chromatography. So the naringenin standard, this one is the naringenin standard. This after methylation we get this product sakuranetin, epigenin standard, and genkwanin. This is the methylated product, and this is the rhamnetin, the methylated product. So we further confirm these compounds by the help of high resolution QTOF ESI mass of the profile of the in vivo reaction of naringenin, epigenin, and quercetin with SOMT. We get the exact mass: 317 for rhamnetin, 285 for genkwanin, and 287 for sakuranetin. So this confirms the production of these compounds, And furthermore, we optimize the production parameters like time, substrate concentration, and the media. So in each case, we could see that at 48 hour of time interval, in each case 48 hour, and when they were supplemented with 200 micromole of the substrate, there was the maximum production of the products. Similarly, we wanted to increase the production, and we use the fermenter for scale up. And here is the large scale bioconversion using fermenter under controlled condition of temperature, pH, and dissolved oxygen in TB media containing E. coli. The experiments were performed in triplicate and repeated 3 times. So here we can see the time of incubation, 48 hours, we could get the maximum production of sakuranetin, almost around 180 micromole. In case of genkwanin also, we could get around almost 175 at 48 hours. In case of rhamnetin, what we saw was during 24 hours of incubation, all the substrate were completely biotransformed into rhamnetin. And we again fed this system with 200 micromole of the substrate quercetin. And finally, after 48 hours, we could produce almost around 370 micromole of rhamnetin. So after getting all these compounds, we further went on to test the anti-cancer activities of these compounds by using the MTT assay. So what you can see here, this is the gastric cancer cell line. This is the melanoma cancer cell line, hepatic cancer and brain tumor cancer cell lines. So this is the sakuranetin. So when the cell growth has been reduced by the help of the product of our compound, here also similarly here also there is a reduction in the growth of the cancer cell line. Similarly here it is comparable, here this is decrease. In case of the epigenin and genkwanin, this dotted line shows the product. The cancer cell lines are decreasing. Similarly here, melanoma, hepatic, brain tumor. In all the cases, the product showed higher activity than the substrate when they were subjected to MTT assay in the presence of various cancer cell lines. Similarly, we tested the effect of sakuranetin and rhamnetin on melanogenesis in B16F10 melanoma cells. The cell viability was determined at 72 hours after compound treatment by trypan blue assay. So you can see here this is the cell viability in 3 micromole, 6 micromole, 12 micromole. There is the cell viability. There is no cell site toxicity. So this is the melanin production. Melanin production has been decreased in case of the product. This black bar shows the product. The melanin production is decreased. Similarly, in case of tyrosinase activity, in case of the product sakuranetin, there is a decrease in the tyrosinase activity. So we can say that the decrease in the melanin production or decrease in the melanin cancer cells are due to the decrease in the tyrosinase activity. That means the sakuranetin acts on the tyrosinase enzyme in case of these cancer cell lines. Similarly, in case of rhamnetin also, we could see that the tyrosinase activity has been decreased in the presence of this methylated product compared to its unmethylated counterpart, quercetin. So the products are having higher biological activity than the parent substrates. Similarly, in case of genkwanin, what we could see is in vitro angiogenesis, the tube formation activity of these compounds we compared. This is the serum-free media. This is the vascular endothelial growth factor containing this endothelial cells. In the presence of the substrate, there is a decrease in the cell concentration. But when we give the product genkwanin, there is almost clearance of these cancer cell lines. Similarly, serum-free media and growth factor containing media. So we can see the tubules. In angiogenesis, there is the rapid invasion by the blood vessels and the tubules are formed rapidly. But when we put epigenin, there is a decrease, and genkwanin, almost clearance of these tubules. VEGF, there is a decrease, this control in the presence of VEGF, and epigenin decrease, and this is the product, more decrease in the invariant cells. and tube formation is rapidly decreased in the presence of our compound, which we produce in our laboratory. So like basically the products which we have produced in the lab condition, these are more potent than the substrate which is commercially available in the market. So after doing the methylation of these compounds, we found that these are more potent, more bioactive, and more stable than the parent compounds. And after doing the methylation, basically it increases the lipophilicity. So we want to increase the solubility of the compound, so it will help in the better formulation of the drugs and could be commercialized. Because the drugs which are more soluble in our body will be taken more easily by our system. So we need to take care of the solubility of the drugs as well. So glycosylation is the tool which can help to increase the solubility of the drugs which are less soluble. So these are the different strategies. Like basically we wanted to attach the rhamnocyte group in the rhamnetin which we have produced. So if this is the rhamnetin and this is the E. coli system, glucose metabolized to glucose 6-phosphate, this pathway has been deleted so that we can channelize this glucose 6-phosphate for the production of rhamnose, TDP rhamnose. And this PGI gene has also been deleted. 2 genes has been deleted in this case. And in the presence of TGS, DH, EPKR, different enzymes, this glucose 6-phosphate is converted to TDPL rhamnose, which when acted by ARGT3, this is Arabidopsis, glycosyltransferase 3, attaches this rhamnose group to this rhamnetin. Similarly, this glucose 6-phosphate is channelized in the presence of Cal S8, S89 enzymes to produce UDP xylose. This UDP xylose, when acted by ARGT3 in the presence of this rhamnetin, will attach to the 3 position of rhamnetin and produce rhamnetin 3-OL xyloside. This is a xyloside form of rhamnetin. Again, for the glycosylation, glucose 1-phosphate, UDP glucose, in the presence of UGT78K1, this is the glycosyl transferase which can transfer the glucose moiety. At the 3rd position of the flavonoid, we produce rhamnetin 3-O glucoside. So 3 different derivatives of rhamnetin were produced: xyloside, rhamnosides, and glucosides. So this is the proton NMR, C13 NMR, COSI, ROSI of rhamnetin. This is the HMBC for the production of the position. So these are the different HPLC which proves the production of detection of the production of rhamnoside, xyloside, and glucoside. And this is the high resolution HPLC, PD acute of ESI mass, which confirm the production of each rhamnetin derivatives. These are the production level of rhamnetin rhamnoside, rhamnetin xyloside, and rhamnetin glucoside by engineered E. coli. So we also tried the in vitro glycosylation by using the enzyme YGIC, which was available in our lab condition. HPLC chromatogram of Niranjan reaction product, sakuranetin. This one is Niranjan, this is sakuranetin, methylated product. And after glycosylation, give rise to 2 peaks, glycosylated peak, glycosylated peak. This is galactosylation, galactosylated peak. Why we doing methylation is to increase the stability and biological activity. Further, we went to do the subsequent glycosylation to increase the solubility. So we will have now more stable compound, more biologically active compound, potent compound, and more soluble compound. So it will be easier to formulate as a drug as a whole. So these are the mass which confirm the production of sakuranetin monoglucoside, diglucoside, monogalactoside, and digalactosides. And the structural elucidation of these compounds are on the process. So coming to the conclusion of this research, taken together, our approach of producing sakuranetin, genkwanin, and rhamnetin facilitated an opportunity to create an economic incentive to develop strains capable of converting cheaper feedstock into high-value compounds. In addition, the compound which is produced using biotransformation provides radioselectivity and chirality, both of which are not easily achieved by conventional chemical synthesis. The results we presented here allowed us to infer that the radioselective methylation of naringenin, epigenin, and quercetin scaffolds may result in interesting derivatives with potent anti-cancer activity, which further deserves further studies, namely regarding their molecular mechanism of action, in order to investigate their potential as anti-tumor agents. So, in conclusion, sakuranetin, genkwanin, and rhamnetin could be produced in industrial scale in future and are very promising agents to be used as anti-cancer, anti-melanogenic, and anti-angiogenic agents, which when further decorated by glycosylation modification could enhance their solubility for drugs formulations. So these are the recent publication by our lab. This is our research group. My professor J.K. Song with his lab members and graduates. Thank you.
Thanks to Dr. Koirala. Is there any question?
Thank you for your presentation.
Thank you.
Just one question.
Yeah.
You are talking about putting on the market the product, something like that.
Mm-hmm.
But did your team assay this product in vivo? In animal model?
No, we have not tried till date. We are on the process of doing that. Till now, we have done only the in vitro analysis of these compounds for anti-cancer assays.
Okay, thank you.
You're welcome.
Thank you very much. I have 5 questions, but this one question it eliminates all of the questions, especially the animal model, because as we know now, there are many miraculous antioxidants and biocompounds.
Yeah.
And the cancer is increasing every day.
Yeah, right.
It's like is some contradiction. So I just because I just want to ask why you choose this animal model, like this worm. Why your team they choose this? Why they isolate this biocompound for silkworm, something like that? Silkworm.
So animal model,
Yes, why they use this worm?
So you mean the compound or the experiment we carried out?
Yeah.
So these compounds?
Because they say here is something like from silkworm.
Silkworm.
Yes. What is this?
I don't think the compounds are isolated from silkworm. These are basically the plant's secondary metabolites.
Is a plant? Is a plant? Okay.
I think you are looking at the different slide.
Okay. This is a melanon cell, right?
Yeah, melanogenic cells, yeah.
It means it's not so difficult to apply in the animal model because, as you know, it's like a skin,
right. Yeah.
It's something like that.
Yeah, we are not able to apply in the animal model because our laboratory condition, we don't have the in vivo experimental setup to carry out the experiments. So we just focus on the in vitro conditions.
Okay. Also, the last question is like you showed the pathway, right?
A pathway, yeah, right.
The pathway. So there's some, because it seems like it's complex. The structure is complex.
Yeah.
Is there some influence during the transportation? Have some special protein to transport the active principle inside the cells, or?
Actually, these flavonoids are taken by the E. coli by the help of the certain enzymes, PGM, phosphoglycosamidase, and kinase enzymes. They help in the conversion of these compounds. And we have used the radioselective glycosyl transferase, which will help to attach the glucose. The UDP glucose is the activated form of glucose molecule, UDP glucose. This will be transferred to the compounds which we have produced attachment of the glucose to the flavonoid will give rise to the glycosylated flavonoids, which will help to increase the solubility of these compounds.
Thank you.
You're welcome. Yeah. Any more questions?